Good morning, everyone. So, what's on my list today? An update on education. Right. Okay. So, basically, how is my education going? Because obviously, I've got my degree starting in October for biomedical sciences. It's a really interesting field, and people are going into it for all different kinds of reasons. Biomedical sciences in general is a very broad spectrum field that has extremely fine detail and generalized areas about it. Because when you're talking about like hyper specifics in terms of medical science, it is very often required to see it in the larger context. For example, I've been doing a little course on epidemiology, so like the study of diseases and how they progress, and things like herd immunity, all of that. But to understand how a disease affects a wider population, you need to understand how it affects an individual, so that measurements you take can be scaled up or passed through equations to estimate proportions of the population that may or may not have been infected, and then kind of using that to inform decisions, such as during COVID. Obviously, all the decisions were very controversial, but politicians had to make decisions based on when lockdowns should occur, and those decisions are largely informed by trials. Is trials the right word for this? Samples, so sampling, testing what proportion of people had antibodies for what variants of the virus over time as well, because obviously variants change, and oftentimes they pick up things that are later on than they should have done, because there was this whole discussion about how COVID was around for longer than people thought it was. But anyway, it's important to see how it affects the individual, so down to things like what types of antibodies are generated on a first response and later responses, IgM antibodies, why are they generated first, what are they, what's their structure, pentamers with J chains, which type of antibodies are more likely to call on the complement system, how is the innate immune system capable of fighting off viruses, what types of receptors inside the cells detect which types of viruses, so which look for double-stranded RNA, for example, they call them PAMPs, pathogen-associated molecular patterns, and how does this stimulate responses to neighboring cells as well, so through interferon, how does this encourage the production of antiviral protein in other cells. Basically, if you didn't know, your body does already have a fairly effective inbuilt defense mechanism, which doesn't even require you generating antibodies for the virus. Because I didn't even really know too much about this before I started doing this course, that you think you're completely defenseless until you have that adaptive immune response and you're generating antibodies and you're fighting it off. But it's not true, and it kind of makes sense because we've evolved on this planet alongside everything else for, well, technically billions of years, right? Although the earth is only like 4 billion years old, but as soon as life was able to start appearing on the planet, it did, which lends credence to an idea that life is actually far more common in the universe than we initially expected. There is an interesting theory about the universe already being seeded in some way with the right kinds of compounds that are precursors for life based on the conditions of the universe in earlier years, where the general temperature of the universe was just higher and the conditions were such that such compounds could have been kind of pre-saved, if you like, in the universe, and therefore scattered along early rock structures that formed planets, and therefore the seeding of life may be more common than people think. But anyway, that's a whole other bag of worms. The point is that we've evolved alongside pathogens on this planet, and therefore we have an intrinsically natural defense against them before the main army kicks in. So that's something that I found quite interesting to learn about. Your body knows what types of RNA structures, not just RNA, but the course focused on single and double-stranded RNA structures. It knows what's not right structurally, and then it tries to break things down. And obviously there are body conditions, genetic conditions that make that less efficient. For example, we learn about TLR3, toll-like receptor 3, inside of endosomes, inside of cells, which can detect things like viral particles before they leave the endosomes and start replicating. But in theory, if someone was deficient in TLR3, then they would have a later detection response for the innate immune system in picking up those viral particles. I might be wrong about some of this stuff in terms of specifics, but that's why I'm learning about it. So why have I been doing this course if the degree doesn't start until October? Well, I wanted to become an expert before it started, and I'm picking fields that I don't have much experience in, because I do have some experience in other fields relating to my own medical problems, and it will be interesting to solidify those. For example, with the inner ear and vestibular system, I consider myself a pseudo-expert on that already, but what I'm lacking is the terminology, the finite anatomical structure that connects the vestibular nerve to the inner ear, and therefore what part of the brain does the vestibular nerve feed into, and on that matter, the neurobiology. I want to ideally be able to have a fully comprehensive mental map of human anatomy inside my mind, and I want to be able to visually zoom into every part of that body and just know how it works. If I was injected with something, I want to know where it's going in the bloodstream. I want to know where it's kind of perfusing into and what effect that's having. on a cellular level. And again, none of this is even necessarily for a job, although it kind of is. So a lot of people that do biomedical sciences are doing it because they want to go for like lab work in the NHS here in the UK. There is something called the Institute of Biomedical Sciences, where it's a little bit hard to get accredited. There are various universities in the UK. I'm not going to tell you exactly what one I'm going to, just for privacy reasons. But when they do biomedical science or biomedical sciences courses, they can either be accredited by this institute or not. And if you're accredited, then it's kind of a bit like a fast track way into starting that lab career. The one I'm doing, and many others, is not accredited by the IBMS, but I knew that before signing up to it. But the thing is, because I'm not going in for a career in lab work, that's not really required. And it doesn't really close off most of the career opportunities that the degree will open anyway. Plus, if you do a degree that's not accredited, this institute, you just contact them, they'll assess the degree, and then they'll just recommend you like top up modules if you ever wanted to go down that path. So the way I'm seeing it is, I'm doing this degree because I want it to, well, make me an expert in a certain area, because I've been fascinated by health science really for a long time, and I just have an immense curiosity about it. But also for my 3D work, like I've said in other videos, I want to open up freelance services for scientific visualization. And this is one of those fields where if you're going to do visualizations for things relating to medical science, you really do need to know the subject to create those visualizations. It is not something you can easily just fake or skim by. And if you're working with a client who wants you to visualize some certain aspects, maybe we're visualizing the interaction between B cells and helper T cells, maybe the interaction between CD40 and CD40L, and telling the B cells to differentiate into different types of cells, like memory cells and plasma cells. Let's see, I'm bringing in what I'm learning from the course. Then you need to know that stuff, because when I was reading about that specific process, it talked about things like expressing this molecule, CD40L, on a cell surface. And when reading language like that, I mean, the biomedical science is such a visual field in my mind. You can just read and learn by text alone, but with specific language like expresses, when a cell expresses a molecule, what does that mean? Physically, literally. What happens? What does the molecule actually do? And that's what I'm interested in, like the actual physical visualization of what's actually happening. If you pass that language on to an artist without the right kind of background knowledge, then there's a potential misinterpretation there. Because what do you think expresses mean? Like, just think about that. If a cell expresses a molecule, what do you see visually when you hear that language? Because the first thing that popped into my mind, which I think would be the first thing that pops into a lot of people's mind, is there's a cell and there's this molecule that goes and kind of just like diffuses off of it. But that's not really what happens, at least not from my understanding. When the molecules express, it just kind of like bubbles to the surface and is ready for contact and interactions with complementary molecules on the surfaces of other cells. So that's the kind of important thing about the intricate learning process, is that if a scientist asked a 3D client to do that, this cell expresses this molecule, and then the artist hands something back where all the molecules that are supposed to be on the surface of a cell just go poof and just disconnect into thin air, then that's kind of funny, but it's not quite right. But also that's just like a communication issue, right? Because even if that knowledge wasn't present, it would be useful if the scientist provided at least like maybe a little storyboard of what should happen in a 3D animation. Just go, oh, well, here's cell, here's molecule. And then while looking at those storyboards, the 3D artist will be able to understand what that language means. So it's really interesting to me actually looking at like the visual aids while I've been doing my course. Because some of them are not particularly high quality, but that's also perfectly great and fine because they don't need to be. What interests me as well is the minimum viable product that's required to convey a piece of information effectively, but I do want to make things pretty, even down to books. Hang on. So here's Biomedical Science lecture notes by Ian Lyons and other people, I think, published by Wiley Blackwell. And in here, there are many diagrams for different things. Now, obviously, they're quite simple diagrammatic, but someone has to make all of these. This is a section on embryology. Okay, we've got general anatomy, and they're quite high quality. They're perfectly just exactly what they need to be to convey the information. Here's a cross-section of the ribs, here's a vein, here's an artery, here's a nerve, and here are the important layers of muscular tissue there to keep in mind. Here's the liver, etc. Now, someone who's just a general scientist and writing all these things, or some kind of consultant or professor or whatever, they don't necessarily have the time to do all those things So I feel like there's always going to be a job for authenticated visual aid creation that is something that cannot necessarily be done by an AI, at least not in this day and age. I mean, it can, but the accuracy checks for it, the authenticity, the certified correct nature of it. So that's where I am at the moment. I'm just about finishing up this virology and epidemiology course, and it's given me like a really good taster into things. Yeah, it was quite information packed. In fact, when I'm done with it, I want to go back through and read it all again because there was a lot of information on like the cell signaling dynamics for detecting viruses. But at the moment, this kind of last stretch has been a lot of talking about herd immunity, basic and effective reproduction rates, ELISA serum studies, and stuff like that, which I find a bit less interesting. I find the actual like the cellular dynamics more interesting, but it's still important stuff to know about. After this, I might see if I can find some information on the hepatic system. So talking about liver and the interactions with the liver and the gallbladder and the pancreas, kind of in this gastrointestinal system, because I feel like that's something where I would benefit quite a lot from having a deeper understanding, particularly for my histamine issues and also for my genetic Gilbert syndrome, but also blood. I'm very interested in hematology as well. So we'll see where to go with that. I mean, at the end of the day, I've got to learn everything, but we'll see how it goes. So anyway, that's education. That's where we'll leave it for now. If you made it this far, put some kind of medical or anatomical related emoji in the comments so I can see if you did make it here. If you want to support my work, my Blender work, my tools development, my studies, you can sign up to my Patreon, patreon.com/CurtisHolt, or check out my Blender tools at curtisholt.online/store. Thank you for watching, everyone. Stay safe, and I'll see you next time.